Hello students, I am Moshimam again here to discuss this the structure of ovary and the various stages of oogenesis. So let us start with our material. Now you see, this is the structure of ovary. This is the internal structure of ovary, and this is ovary. We have already learned the location, about the location of the ovary. The ovary is located in the lower abdomen, and it is about 2 to 4 centimeter in length and about 1.5 to 2 centimeter in breadth, and it is attached to the dorsal surface by the peritoneal covering called mesovarium, and it's attached with the uterus by the ligaments. So let us have a detailed structure of the internal structure of the ovary. Now you see, this is the mesovarium. Inner to mesovarium, this is the germinal epithelium. This germinal epithelium is the basic structure which give rise to oogonial cells, or finally the oogenesis occurs from which cells, those cells. And inner to germinal epithelium, there is another layer called tunic albuginea. And if we go on inside the ovary, we will find that there are two distinct parts. This one is the cortex, the outer part, which is richly supplied with follicular cells and which contains follicular cells and richly supplied with connective tissues. And here is the inner part, the medulla, which is richly supplied with blood vessels. Now, first of all, let us consider this. The oogonial cells, larger oogonial cells, larger germinal epithelial cells divides and redivides to form numerous oogonial cells, clear, which forms the egg nest. From these oogonial cells, the oogonial cells divides and forms the primary oocyte. You can see here this primary oocyte. When this primary oocyte gets surrounded by single layer of follicular cells, it is called primary follicle. So it is the primary follicle. Next, this primary follicle, the primary oocyte, starts to divide, and it undergoes meiosis 1, but it cannot complete meiosis 1. This meiosis 1 get arrested at its diakinesis phase. The whole oogonial cell, this proliferation, formation of oogonial cell, primary oocyte cell, occurs in the fetal state. Now this division get arrested, and in the onset of puberty, each of the follicle starts on undergoing division in each 28 days ovarian cycle. So next, what happens? This oocyte now get encircled with numerous layer of follicular cells. And the thicker layer also appears. This follicular cells comes from the germinal epithelium only, but the thicker layer comes from the cortex. So this structure is now called secondary follicle. Now, after this, the secondary follicle further develops. The thicker layer increases. It forms two distinct layers, theca externa, theca interna. The follicular cells get stratified, releases liquid, which forms the liquid folliculi, and it gets they actually form some initial phase of the antrum, and these which are filled up with the liquid folliculi. And thicker layer also increases lot, and now the primary which had started to divide at the onset of puberty, when this tertiary, so this is the tertiary follicle. With the formation of tertiary follicle, these primary oocyte completes its division and forms the secondary oocyte and one polar body. So secondary oocyte and one polar body. And finally, you can see here the whole thing, this fluid filled area enlarges. More or less the whole surface of the oocyte is covered, secondary oocyte is covered with the fluid filled area, and there are layers of granulosa cells also, and this the major layer of granulosa cells is separated from this the granulosa layers of granulosa cells by this fluid filled area. But the region where this antrum is absent and this layer of granulosa cells is considered with continuous with the secondary oocyte, this is called cumulus ooferus. So this is antrum, distinct antrum is formed, cumulus ooferus is formed, several granulosa cells, several layers of granulosa cells, several layers of thecal cells clear, and this is the secondary oocyte, and there is one polar body, and the secondary oocyte is covered with vitelline membrane, zona pellucida and corona radiata, and so now it is the matured Graafian follicle. This matured Graafian follicle finally ruptures, and the secondary oocyte which has been formed is released, and we know that it is released in the fallopian tube. It moves into the fallopian tube and goes to the ampullar rhythmic junction and rests there for 48 hours. But the ruptured Graafian follicle now releases certain ruptured Graafian follicle forms the corpus. It releases yellow pigment, carotenoid pigment, and forms corpus, which are lutein, and forms the corpus luteum. The corpus luteum now enlarges and forms mature corpus luteum and secretes progesterone. Now, if there is fertilization, it is maintained, but if there is no fertilization, it gradually degenerates and finally forms corpus albicans. This ovary also releases another hormone we know, that is the estrogen, and it is secreted from these granulosa cells. Next, we will discuss another very vital part of the female reproductive system, that is the oogenesis, that is the formation of the egg. Now you can see here, these are the oogonial cell. These oogonial cells, which are formed from the germinal epithelium, this oogonial cell divides and redivides to form numerous oogonial cell, which is called this phase is called multiplicative phase. Now this numerous oogonial cell now undergoes mitotic division and proliferates to form primary oocyte, and this is called growth phase. This proliferated primary oocyte formation is called growth phase. Now this primary oocyte, so all this part occurs in the fetal state. In this fetal state, this primary oocyte starts dividing, and the primary oocyte starts dividing to form, undergoes meiosis 1, but it cannot complete meiosis 1. It gets arrested in the diakinesis phase, and now at this phase, after the birth occurs, and after the birth it continues till puberty, it remains as such. And at the onset of puberty, in each ovary, one of this primary oocyte completes it meiosis 1 alternatively. So one of the primary oocyte completes its meiosis 1 alternatively and forms a secondary oocyte and the polar body. The secondary oocyte is the functional one, which will form the egg. It contains the cytoplasm, it contains the chromosome, but the polar body contains very less amount of cytoplasm, but the chromosome number remains the same as the secondary oocyte. So the chromosome number is halved, and the chromosome number of the polar body is similar to that means if the primary oocyte is having 46, it will have 23. So 23 chromosome is there in the secondary oocyte, 23 chromosome is there in the polar body, but cytoplasmic part is mainly in the secondary oocyte. The polar body has the least amount of cytoplasm. Now this secondary oocyte will start dividing, and this secondary oocyte will start dividing, but it will undergo meiosis 2, but it get arrested at metaphase. It forms metaphase promoting complex and get arrested at metaphase. Now at this stage it is released from the ovary and it moves into the fallopian tube and it waits here for 48 hours. Now during this phase, if the sperm enters, when the sperm enters, these metaphase promoting complexes are destroyed and anaphase promoting complex is produced, which so the meiosis 2 is resumed and it forms the ovum and the second polar body. So meiosis 2 continues completed and it forms the ovum and the second polar body. And this phase is called the maturation phase. So this phase, so if we consider the location, till this it occurs in the ovary, but after this, that from secondary oocyte to ovum formation, that occurs in the fallopian tube. And from oogonia to primary oocyte, that is, it occurs in the fetal state. Next, it occurs in this phase occurs after the child reaches the puberty. So this is about oogenesis. Go through it.